In this lab, we will test the water quality of various samples collected throughout the area. The samples are tap water, wetland water, canal water, seawater, and deionized water for a control. We will begin with canal water. The first test we will conduct is for nitrogen. Nitrogen is one of the most abundant elements on Earth. 80% of the air we breathe is nitrogen. It is found in the cells of all living things and is a major component of proteins. However, too much nitrogen in aquatic systems can deplete the oxygen levels in the system, thus killing off aquatic organisms. Nitrogen pollution of our water systems is caused by poorly operated water treatment plants, sewage leaks, and agricultural runoff. There are two ways to test for nitrogen. The first is by a test kit. Fill a test tube to the 5 milliliter line with the water sample. Then add a nitrogen number 1 tablet. Cap the tube and mix until the tablet is entirely dissolved. Then add 1 nitrogen number 2 tablet. Cap and mix again until the tablet is dissolved. Then set the tube aside for 5 minutes. While you are waiting, go ahead and repeat the steps for a sample of DI water. Remember, this is your control. Once 5 minutes has elapsed, place the test tube into the OctaSlide viewer. Match the color of the sample with the color standard. Record this number as the amount of nitrogen in ppm, or parts per million, in your sample. As you can see here, the value is approximately 10 ppm. The other method is simpler. Here we will use prepared test strips. Take one test strip, being very careful not to touch the test area with your fingers. Dip it into the water sample for 2 seconds. Remove and wait for 1 minute. Then match the test area with the provided chart. Record the ppm. The next parameter we will test for is phosphorus, an important element for the growth of plants and animals. However, too much phosphorus in an aquatic system can lead to algal growth, which will deplete oxygen from the system and kill the system. Begin by filling a test tube to the 10 milliliter line with your sample of water. Use a pipette to then add 1 milliliter of phosphate acid reagent. Cap and mix well. Then add 0.1 grams of phosphate reducing agent using the provided spoon. Cap and mix until the powder is dissolved. Then place the test tube aside for 5 minutes. Again, while you are waiting, go ahead and repeat these steps for a sample of DI water. Once 5 minutes has elapsed, remove the cap and place the test tube into the phosphate comparator to match your sample color with the color standard. Record this number as the amount of phosphorus present in your sample in ppm. Next, we will test for hardness. This simply refers to the amount of calcium and magnesium ions in the water. These minerals enter the water system mainly by the leaching of rocks and soil. Calcium is an important component of both aquatic plant cell walls and the shells and bones of aquatic organisms, while magnesium is an essential nutrient as well as a component of chlorophyll. There are two methods used in this lab to test for hardness. The first involves a test kit. For any freshwater sample, such as canal water, fill the test tube to the 50 milliliter line and add the hardness test tablets one at a time, dissolving each completely before adding the next. Stop adding tablets when the water has changed from red to blue. Then, multiply the number of tablets used by 40 and record this number as the ppm total hardness. If you are testing a sample of seawater, pipette 2.5 milliliters of your sample into the test tube and dilute up to 50 with DI water. Add the tablets one at a time until the color changes. Then, multiply the number of tablets used by 800 to obtain your ppm of total hardness. The second method is again test strips. Dip a test strip into the sample for 3 seconds. Remove and gently blot the excess water on a paper towel. Then, match the color of the test area with the provided chart and record your ppm. The final series of tests will be for pH and alkalinity. pH refers to how basic or acidic a system is, while alkalinity simply refers to how basic a system is. All living organisms have an optimal pH range at which they live. Anything beyond that range, and the organism will die. To test for pH, use the pH indicator paper. Simply take one strip and dip the test area into the water sample. Remove and blot the excess water. Then, match the colors with the provided chart. To test for pH and alkalinity, take one of the test strips and dip it into the water sample for 10 seconds, trying not to agitate the water. Remove, blot, and immediately match the pH color. Then, match for alkalinity. Record your results. Here we have two samples of wetland water. As you can see, one contains aquatic vegetation and one does not. In class, we will discuss the functions of a wetland, one of which is to filter out pollutants from the system. Given this information, which sample will contain higher amounts of nitrogen and phosphorus? The sample with vegetation or the sample without?